Our first afternoon session, we have Tony Glimp Martin, and she's an environmental microbiologist specializing in water and wastewater treatment and a treatment process consultant with Wastewater Microbiology Solutions. She provides technical assistance, consulting services, and microscopic evaluations for more than 30 industries in 80 municipalities. Prior to that, Tony served as a senior level supervisor in the Monitoring and Research Division of the Metropolitan Water Reclamation District of Greater Chicago, and has more than 40 years of water quality related experience. Tony has conducted over 400 technical workshops and has made numerous presentations at conferences, public meetings, the Great Lakes Commission meeting, and for state representatives. We're really lucky to have Tony. I got to watch her presentation last year. This is going to be awesome. Please join me in welcoming Tony.
Thank you. Enzymes are very efficient proteins. They trigger biochemical reactions in living organisms. Under favorable conditions, they can increase the rate of biochemical reactions 10 billion times more rapidly without the enzymes. But their function can be altered with changes in the environment. So if you have changes in nutrient or temperature, pH, or some toxic chemicals come in, it can affect the enzyme function. So without enzymes, they can't break down organic material, and they can't even perform their cellular processes. And they only do what they can do when the conditions are right. So you need to understand that enzymes are substance specific. So if somebody comes and tell you that they've got these super enzymes that can do this great work, you need to understand that they are substance specific. So you have one enzyme, proteinase, to break down protein. You have one enzyme, lipase, to break down fats or lipids. You have an enzyme, glucuronidase, that breaks down carbohydrates. And so if you're gonna get some enzymes, you better get one for every single thing that's coming into your treatment process, because they're very substance specific. They are useful though. They're very useful if you've got a difficult to treat waste. So if you've got a meat packing industry down the street and they're sending in all this protein, then you can get some help with enzymes to help with that difficult to treat waste like proteins. If you got a whole bunch of fat in restaurants and high density restaurants in the area and you just have a lot of fat coming in, then by all means get some help breaking down that difficult to treat waste. But most systems, you don't really need to have supplement enzymes unless you have a difficult to treat waste stream. Okay, so if your enzymes aren't working, then their enzymes aren't going to work. So we need to find out why your enzymes are not working under normal conditions. Okay, so I want to just talk a little bit about the phases of the growth of bacteria, because they mimic this phase in the treatment process. So I want you to understand these different phases. So it's the lag phase, the accelerated growth phase, the declining growth phase, the stationary growth phase, and then there's the death phase. So the lag phase is kind of at the beginning. At that beginning, like if this is the aeration basin, at the beginning of the aeration basin, the food level is high. By the time the wastewater flows through the aeration basin, the food level should be low. At the beginning, the bacterial level should be low, but by the time the bacteria consume the organic material, they should increase. So early in the process, there's a lag phase. And during this lag phase, there's no growth, there's no reproduction. So bacteria don't automatically just start eating organic material because they come in contact with it. These lag phase can last up to 20 minutes and sometimes several days. And so bacteria don't just instantly start consuming organic material. They have to be acclimated to the environment. They have to produce the necessary enzymes. That's another reason why I like primary, because what primary does is it stabilizes that load. It acts like an equalization basin. So it gives you a sort of a stable loading coming in. So the bacteria are not going, oh, today we got protein. Oh, tomorrow we got fats. Or so they're not having to keep producing different enzymes and you're driving them crazy. Okay. So then once they've developed all the enzymes that they need, then there's this exponential accelerated growth. They grow super fast. They grow at the fastest rate possible. They consume the organic material. They're constantly dividing. They're doubling exponentially. the cells are active and the food is reduced at the highest rate. So all they're doing is they're splitting together, they're eating and they're splitting into two, eating and splitting into two. Somebody asked me, how long does a bacterium live? I said, forever, because all he does is he eats, splits, eats and splits, eats and splits. As long as there's lots of food available, that's all they're going to do. They're going to eat and split, eat and split. Even your flat formers, that's all they're going to do is eat and swim and split. And so the bacteria are modal. They swim around. They have a tail like this image you see on the side. They have a flagellum. As long as you give them food to eat, they're going to just do nothing but split into two identical cells and multiply. Kind of like us. If we just keep eating, we're going to double too. Unfortunately, we don't split in two. But as long as that food is there, that's all they're going to do is swim around and divide. But then comes the declining growth phase. And during this phase, most organic material is removed now. And so now you got more bacteria than you have food. So reproduction begins to slow down. Then your next phase is your stationary phase. And during this phase, the number of bacteria is relatively constant. And so your bacteria are going to take some steps to conserve energy. They say to themselves, and I've heard them say this, they said, look, why are we swimming around like crazy, wasting energy? Let's just get rid of these tails and let's conserve our energy. And just, they developed this thick outer slime layer. And they said, let's just stick together and help each other out. And that's how they begin to form the flock, okay? So now, if you keep on going, you get to the death phase. And at this phase, the number of deaths exceeds the number of new cells. Most of the food is depleted. You might start getting some toxic waste products from the death and dying microorganisms, and it can create an unfavorable environment. So we don't want to leave them in there too long. So if this is my aeration basin, when the wastewater comes into the aeration basin, at the beginning of the aeration basin, you'll see the food level is pretty high. By the time it gets to the end of the aeration basin, the food level is low. At the beginning, when the return sludge comes, when they bring in the microbes in, the bacteria are very active. They're swimming, they're active, they're just splitting into two identical cells. But towards the end of the basin, they're conserving energy, they get rid of those tails, they clump together, and they form flock. So you need to understand that it's important that we leave these microbes in this basin long enough to consume most of the food so that they can clump together to form flock. At the beginning, early in the process, because food is abundant and available, they're growing, they're modal, they're extremely active. As long as enough nutrients are available, bacteria will use the nutrients mostly for growth, and they'll multiply very rapidly, splitting into identical cells. As time passes and the food level decreases, the bacteria shed their flagellum to conserve energy. They begin accumulating a thickened slime layer that enables them to stick together to form flock. So if you kind of look at this for an example, the food level is high, your bacteria are just single little cells splitting into identical cells. But as the food level becomes low, that's when they begin to clump together to form flock. The important thing is that the bacteria must remain in the aeration basin long enough to remove most of the food. Because it's only when the level of food is low that the bacteria form that thick outer slime layer that allows them to stick together to form the flock. Now, if this is my aeration basin and this is time, what would happen if I shortened my aeration basin, the time in my aeration basin? What would happen? The bacteria are just going to be splitting into two. They're going to be splitting into identical cells because the food level is still high. So the time in this tank is important. Okay, so there's several factors that affect the bacteria in the treatment system. The sludge age, which is what we just talked about, how long we allow them to be in contact with the food, the temperature, the amount of DO, mixing, pH, and nutrients. All of these affect the bacteria in the activated sludge system. We talked about sludge age. Sludge age is how long the solids containing bacteria remain in that system. Early in the process, when there's lots of food, they're active, they're dispersed. But as the sludge ages, they lose those little tails, they accumulate a thick layer, and they clump together to form fly. Temperature affects them. Biochemical reactions are temperature dependent. Reactions are slower when it's cold. When it's warmer, they move faster. It takes more bugs to do the work in the wintertime than it does to do it in the summertime. But I have to caution operators. A lot of times what they'll do, when I worked for the Metropolitan Water Reclamation District, we have 7 water reclamation plants. And one of their plants was having problems, and I said, well, give me a sample and let me take a look at it. I took a look at it, and I said, well, you have too many bugs and not enough food. And he said, well, it's wintertime. At wintertime, we always increase the amount of bugs. And so a lot of times what operators will do is they'll just follow these rules of thumb without really understanding why we're following these rules of thumb. So I looked at I also looked at the loading that was coming into his plant. And so I noticed that in the wintertime, his loading went down. So if your loading is constant and you have this many bugs, in the wintertime, you will take more bugs to do that same amount of work. But if your work decreased, you didn't have to increase your bugs. So don't just increase your bugs to increase your bugs. Just understand that it takes more bugs to do the same amount of work. But if the work decreases, you don't have to increase your bugs. And that's what they were doing. They had less work and then they had increased their bugs. And so it was extremely low F/M conditions. Oxygen. Bacteria require 0.1 to 0.3 milligrams per liter of oxygen to function. But you need to understand that flock is made up of a bunch of bacteria clumped together. So you wonder, why do we have to maintain 1 to 2 milligrams per liter in the bulk fluid? Well, because you need to have enough oxygen to penetrate into that flock to give the ones on the inside some oxygen too. Because if you don't have enough DO reaching the center of the flock, the bacteria in the center will die. And then your flock solids can start to break up. And so, and you can develop some low DO filamentous bacteria within those flocks. Mixing is required. Why is mixing important? Because they got wet of their tails. And so now you just got a bunch of fat bugs that are just sitting there and they can't do anything. So you got to mix them so that they can bump into each other so that they can form flock. If you don't have good mixing, you're not going to have good treatment. pH affects them because pH affects the enzymes. Optimal pH is 7 to 7.5. They can work outside of those ranges, but rapid pH changes should be avoided. That's why I like primary or some kind of equalization so that the bugs aren't exposed to changing conditions all the time. Nutrients, just like most organisms, bacteria require nutrients to grow and maintain their cell function. They need carbon, nitrogen, phosphorus, sulfur, metal, ions, magnesium, calcium. Most municipal wastewaters contain the things that they need. It's generally systems that are industrial systems or municipal systems that have a significant industrial input can lack some of these nutrients. And we'll talk a little bit later about what happens when they don't have their nutrients. There's this ideal formula for nutritional needs of microorganisms. And this is ratio of BOD to nitrogen to phosphorus. And they say it's 100 to 5 to 1, but I say it's about 100, somewhere in the range of 5 to 10 to 1. And if any of these nutrients are lacking, there could be problems. So what this means is that for every 100 milligram per liter of food that these bugs have to consume, or organics they have to consume, they need 5 to 10 milligrams per liter of nitrogen, and they need 1 milligram per liter of phosphorus. So they need this much nutrients to do that much work. So if they don't have these nutrients, they'll still kind of do that work, kind of like us. We can still function even if we don't have the right vitamins. Our hair might fall out, our teeth might fall out, but we still be walking around hairless and toothless. But that's kind of what happens to the bacteria. When they don't have their proper nutrients, they might still remove your BOD, but you're going to get some byproducts that's going to interfere with your treatment process. Trying to satisfy hundreds of millions of bacteria seem impossible. They got 3 essential needs. They need the right amount of food, they need optimal environment like DO and nutrients and mixing, and they need enough time to do the work. And this other concept of food to microorganism ratio, a lot of times operators will operate their plants maybe trying to maintain a certain sludge age or trying to maintain a certain mixed liquid concentration. But food to microorganism ratio is a critical tool, and it's one that we really need to pay attention to as operators, particularly this one affects the microbes more than any of them. And food to microorganism ratio is simply the amount of food you have compared to the amount of bugs you have. It's a valuable tool for monitoring, it's a valuable tool for troubleshooting. And matter of fact, it's one of the most valuable tools. And it's the calculated mass of the loading over the calculated mass of the bugs. And the concept is to match the amount of food with the organic loading. So I'm gonna give you an example. My ex-husband and I have 2 sons. So my ex-husband and I and my 2 sons, we had just enough food in our house for us. Okay, just enough for us. My in-laws came to live with us. His brother, his brother's wife, their 2 kids, the niece that was living with them, she had a kid and the niece was pregnant. So that was 6.5 more people came to live in our house and they didn't bring food. So that means that we had extremely low food to people ratio. Okay. And so you had 2 choices. We could either go out and get an extra job and bring in enough food to feed everybody, or we could waste out some people. So you can imagine what my option was. Okay. And this right here are my in-laws. Okay. So when you the concept is, say for instance, if influx of food came into my aeration basin, all of a sudden I've got this increased load of food coming into my aeration basin. What do I do? How do I get more bugs to match this food? And I can't tell you how many operators who I've come across who think, well, we'll just increase this return and we'll bring more bugs in here faster. That's not what we want to do, because if we bring more bugs in faster, then they're going to come out of here faster. So the time that they spend in there is going to be shorter. But when you bring in more food, you want that time to be longer. So the way to bring in more bugs or to increase your bugs is not by returning more bugs faster. The way to increase your bugs is through your wasting. And so what wasting does is it can siphon off the concentration of bugs that's going in here. So if I decrease how much I'm wasting, then I'm increasing the concentration that's going into this aeration basin. Okay, so I won't go over the calculation, but just understand that actively growing bacteria can consume food and utilize oxygen at a rapid rate. So if you got a high oxygen uptake rate, then that's associated with a high F to M. If you got a lower uptake rate, then that's associated with a lower F to M. The main concern, though, is not if you use BOD or COD or TOC for food, or even if you use mixed liquid suspended solids or volatile suspended solids for microorganisms. The critical thing is that you always do it the same way all the time. People have asked me, what is the right F to M ratio for my plant? I'm telling you, figure that out. You have to figure that out for your plant. And we can't just always be looking at the manual and say, this is what the book says, because it depends on the kind of waste that's coming in. If it's readily assimilable, if it's easily dissolved, or if they got to crack open the pecans in the shell. So you figure out what your number is. But the most important thing is, however you do it, always do it the same way all the time. Because we're looking for trends here. Because changes in F to M will cause a change, or it can upset your biological activity in your plant. Okay, so that's bacteria, but bacteria are boring for a microbiologist. They're little, they're small, they got little itty tiny tails. So I like to look at the protozoa, because the protozoa give me an idea of what's going on with my bacteria. So since bacteria make up 95% of the microorganisms, they are primarily responsible for moving the organic waste. Your protozoa, they don't care about the organic waste. They like to eat the bacteria. That's their chocolate. So although they do very little, they do play a critical role. Because remember, all kinds of bacteria come into this treatment process. All of them do not have the ability to form slop. So you're going to still have a bunch of bacteria hanging out in that bulk fluid. And so the role of protozoa is to swim around and eat up all of that extra bacteria. So they do an excellent job of helping to clarify your effluent. Most of them are aerobic. There are some that will take soluble nutrients like the bacteria, but most of the protozoa have specialized structures or mouths that they can feed on other microorganisms or whatever fits into their mouth. So this classification, we like to classify them based on how they move. You have your amoebas, your flagellates, and your ciliates. And then your ciliates fall into three categories. Your free-swimming ciliates, your crawling ciliates, and then your sessile ciliates. Those are ones that just stand in one spot. So let's talk about the protozoa. So this is my aeration basin. The food level is high at the beginning. The bacterial level is low at the beginning. So the first ones that start showing up on the scene are my amoeba. Amoeba are the most primitive protozoans. They feed on small organic particulates. They'll eat anything that stands still, and they love the pecans in the shell. Okay. So they don't contribute much at all to the treatment process because they move so slow, they can't compete with other bacteria, they can't compete. They can only dominate early in the treatment process. So they're good indicators. If I had a banquet and I sent an amoeba person and a speed racer person to run down and get the food, of course, the speed racer person is going to get there first. If it's a whole lot of food, then by the time the amoeba gets there, he'll eat. But if it's only a little bit of food, by the time the amoeba person gets there, it's going to be all gone. So the only way amoebas can survive is if there's a whole lot of food in the treatment system. So here's an example of amoeba. They contribute very little to the treatment process. They move very slowly and they can't compete. And so they will take and they will envelop whatever they think they could eat. And then they'll secrete enzymes and then they'll break it down and then they'll eat it. It's a slow process. So here's an example of an amoeba. This is how fast they really move. I have two sons. If you move that fast in my house, you will not eat. You have to move a whole lot faster than that to get to the biscuits. So that's one example of an amoeba. Here's another one. Amoeba have like a sticky coating on them, so that things like they can't really move around that much. And so when something comes and bumps into it, it kind of sticks to it. And then that way they can kind of blob around it and feed off of it. But it's a slow process. So the chances of them eating anything is very, very low if there's not a lot of stuff in the fluid to bump into them. So it has to be a lot of stuff available in order for them to survive. Here's another example. And then one more example. This is the speeder one. This is the fast one. So he just kind of blobs through the water and feeds off of whatever sticks to him. So if I grab my sample from the discharge end of my aeration basin, and I see amoeba dominating in my treatments, in my sample, that tells me something. First of all, you need to understand, if you grab a sample, you might see some of all of the ones that I'm going to talk about, but it's the ones that's dominating, the ones that's the most dominant, that tells you what that environment, what the conditions are like. So if I'm grabbing samples from the end of my aeration basin where most of the food is supposed to be gone, and if I see a bunch of amoeba, if that's what's dominating in my treatment process, then something's wrong. That means maybe a shock load came in, or maybe there's lots of particulates, because amoeba like the pecans in the shell. After a heavy rain, you might see an increase in amoeba. It can also mean a lack of oxygen, because you see how slow it moves, so it doesn't take a lot of oxygen. Some of the other protozoa move a whole lot faster, they require more oxygen. This one, if the oxygen level is low, those other ones might die off, but your amoeba might still continue to live. It can also mean low levels of toxicity or unfavorable conditions, because some amoeba have the ability to protect themselves under those types of conditions. Okay, so now the food level is starting to go down a little bit. Your bacteria is getting acclimated, it's gone to their little lag phase, and they're starting to create the little enzymes and the things that they need. The next thing that shows up on the scene are your flagellates. Flagellates are, and bacteria both like soluble organic material. So these flagellates are competing with the bacteria. So it can only compete early in the process while your bacteria is still kind of going through the acclimation period. Because think about it, flagellates multiply every 5.5 hours. Bacteria multiply every 20 minutes. So by the time one flagellate splits in two, one bacteria would have turned from 1 to like 260-something thousand. So they can't compete. Once the bacteria takes that exponential growth, then they're taking up all the soluble organic material, and then your flagellates can't compete. So most flagellates absorb nutrients just like the bacteria. They compete with the bacteria for dissolved nutrients, and they peak in the system when soluble concentrations are high. They come in a variety of sizes and shape. They have that whip-like structure called the flagellum, and they use it for locomotion, and they use it for feeding. It's like a straw. They can just suck water in and absorb the nutrients. So here's an example. They come big, they come small. Those little tiny dots that you see there, that's bacteria. That's why that's so boring. The little squigglies, that's bacteria. Your flagellates can be big ones, or they can be small ones. And all of this stuff on the side, that's flock. So that they can be big or small. It doesn't matter which one it is, it's just different species that mean the same thing. So if I grab a sample from the discharge into my aeration basin where most of the organic material is supposed to be gone, and I see a bunch of flagellates, if that's what's dominating in my system, that should tell me something. That tells me that I got incomplete treatment, that I still got lots of soluble organic material, and I need more time in that tank. Or it could mean a shock load came in. Another thing it can mean is that there's lots of dead or decaying material, because flagellates are what's called saprophytic. That means that they like to feed off of the juices of dead things. And so what they'll do, if something comes in and kills some of the higher life forms or the bigger microbes, then what they'll do is they'll take that little straw and stick it inside their body and suck off of their body juices. So they feed off the body juices. Okay, so the next one starts showing up on the scene are your ciliates. And we classify them in 3 ways: as free-swimming ones, crawling ones, or sessile ones. So the first ones that show up on the scene are your free-swimming ciliates. So now your bacteria have exponentially grown. You got a whole bunch of bacteria. They took off in that exponential growth, the accelerated growth phase that we were talking about. Now you got all of these bacteria. There are a bunch of them swimming around all over the place. And so your free-swimming ciliates show up on the scene because they can swim fast too. They swim through the fluid. They're chasing all that bacteria. That's their chocolate. So they're chasing after them, and they're feeding off of all of that bacteria. So as your flagellates begin to disappear, your bacterial population increases. There's lots of dispersed bacteria. Free-swimming ciliates begin to dominate because they feed on that increased bacteria. So here's some examples. There's lots of different species. This one is called Nematodes. You don't have to know their names. Just kind of look at their behavior, see how it just kind of, this one always hangs out in the fluid. Then there's a big old Paramecium. That's a Paramecium. But all while they're traveling, they're sucking in fluid and sucking in bacteria. Here's another example. This one kind of reminds you of that vacuum cleaner, the Roomba. And then a few more examples. These are all free-swimming ciliates. And if you watch this one closely, you can actually see him pulling the fluid in, and he's pulling all those little dots. That's bacteria. So he do a really good job of just kind of swimming through that fluid and clarifying that effluent. So if I'm grabbing samples from the discharge into my aeration basin and I see free-swimming ciliates dominating in that system, what does that tell me? That tells me that I must have had a whole bunch of bacteria. And if I've had a whole bunch of bacteria, that means that they multiplied because they ate a whole bunch of food. So that means most of the organic material is gone. But that's not all I want. I don't just want all of the organic material gone. I want it to be so low that the bacteria start clumping together to form flock. So it's still a little bit early. Even though most of the organic material is gone, it's not good enough yet. I still want it to be so low that the bacteria take those steps to conserve energy so that they can form flock. Because the key to good treatment is that the formation of flock, that separation of the solids from the liquids. Okay, so now the free-swimming ciliates have eaten up a lot of the bacteria that's left in the fluid. Your bacteria levels are starting to go down. Your food levels are starting to go down. There's not much left now, so now your bacteria is starting to form flock. And so the bacteria that's formed in the flock, these free swimmers, they can't eat it. So the next ones that start showing up on the scene are I call them the dial bathroom cleaning scrubbing bubbles, but these are your crawling ciliates. So your crawling ciliates, they can't swim. So they can just spin around until they bump into something and they walk. So they use their cilia as legs, and they crawl on the surfaces of solids. And so if you see in the picture, they just have these little legs that help them to crawl. So they don't swim around in the fluid, they crawl around on solid particles. So they kind of look like this. It's several different species. It doesn't matter. It's a crawling cilia. So if I'm grabbing samples from the discharge into my aeration basin and I see lots of crawling ciliates in my sample, that's a good sign. That means I have flock. And the reason why I have flock is because most of the organic material is gone and the bacteria is forming flock. And so that means I expect to get good separation between my liquids and the solids. So that means that time in that tank is adequate. But we never stop at adequate. We have to nitrify, we have to go a little bit longer. So now it becomes the survival of the fittest. So the next ones that start showing up on the scene are your sessile-ciliates. These are ones that have stalks, and some of them, these are ones that stand in one spot. So some have stalks, and some don't have stalks, but they still stand in one spot. And those are your attached ciliates. So the first ones that show up are your stalk ciliates. Now this guy can outcompete all the rest of them because he can stand in one spot. He can rotate his cilia on the top of his head, and he can suck food, create a vortex, and suck food into his head. So he can outcompete any of them. So you guys, if it's one of you guys that was in there that could take and just suck the food from the restaurant into his head, and the rest of you guys had to get up and get in your car and drive, he'd have the advantage. So he can just sit there and just constantly suck fluid in and get the bacteria. So that's my 30-year-old son, Daniel. He sits on the sofa. I'm in the kitchen. The refrigerator door flies open. I see a chicken leg flying out, and I follow that chicken leg all the way into the living room, and there it is, Daniel, sitting on the sofa, and it gets sucked right into his head, okay? So that's Daniel. They feed mostly on suspended bacteria, algae, smaller protozoa, anything that can fit into their head. So anything Daniel can eat will go into anything that goes into his mouth, he will eat it. So they use their cilia to create a current to bring the food particles to his mouth. So it's kind of like this. Some of them have a muscle fiber, some of them don't, but this one, he can just bring the food right to his mouth. Now this is Daniel in action. This is him sitting on the sofa. And that's the refrigerator door over there. And you can see this whole train of food leaving and headed right into his head. Okay, that's my son. Okay, so now it's still not much left because Daniel ate just about everything that's there. So the next guys that start showing up on the scene are your colonial stocks. These guys have multiple heads. They have more than one head on their stalks. That's my oldest son, Lawrence. And when Lawrence is home, Daniel doesn't eat. And when Lawrence and Daniel are home, I don't eat. I'm like the amoeba. I'm the amoeba, okay. It's got to be a whole lot of food in the house if I'm going to eat. So some of them grow in single stalks while others can branch in colonies of more than 100 heads. Some have a muscle fiber, some of them don't. It doesn't mean anything. So this is Epistylus. It's a colonial stalk ciliate, multiple heads. Cartesium. You can see how many heads you get. You see how old this sludge looks. You should never let your sludge stay in there that long where you have all these heads. So here's an example of colonial. Now for the most part, all these mean is that the sludge is older. This is the only one that you should really be a little bit concerned about, because if you notice this one, if you look at the tops of the heads on here, they look like they have a cap. So it has like a cap. And so this one has the ability to close his mouth. Daniel, on the other hand, no matter what it is, it will go in. If it's good for him, if it's bad for him, it could kill him. They don't care. He just goes in. This guy can decide, no, I don't quite like that. And so he has the ability to close his mouth. And so you'll see this one in wastewaters that are a little bit more dirty, where the other stalk ciliates might die because they don't know how to not eat something. This guy can say, I'll survive because if it's not good, I'll just close my head up for a while and wait until things get better. So if you kind of watch I don't know if you guys can see my pointer, but if you watch the guy, the second one from the left, you'll watch him close his head. Okay, so if I grab a sample from a discharge into my aeration basin and I see lots of stalk ciliates, that means most of the organic material has been removed. As the sludge ages, the dominance of stalk ciliates changes from single stalks to colonial stalks. The greater number of heads, the older the sludge. Just remember that. The more heads, the older the sludge. So now not much left. So not much is left. So you start seeing more dominant types coming. So it's like really the survival of the fittest. So you'll start seeing stentors and suctorians. This is a stentor. This guy can outcompete Daniel and Lawrence because Daniel and Lawrence can only fill up their heads. This guy can fill up his whole body. Okay. So he can get a whole lot more fluid in, and it's not that much left in the fluid, so he has a better chance of getting the little bit of bacteria or whatever's left. So if you look at this, that person that in the background, that's my baby Daniel back there. You don't have a chance when this guy is home. This is my oldest stepson. So when he's home, my poor baby don't have a chance in the background. You should never, ever have a colony of these in your treatment system. It is really means it's really, really old. See how black that sludge is? That means you should have wasted a long time ago. So here's a colony of them. I have it at a lower power. But if you can just look at it, look how bad the fluid looks. Look how murky and dirty the fluid looks. This means it's just so old. Everything is dying. All the flock is breaking apart. You should never, ever see stentors dominating in your treatment system. Okay, nothing's left now. So what do? If there's no food left, there's no bacteria to eat, what do? They start eating each other. So the next one to start showing up on the scene are suctorians. Now suctorians, what she does is, you see that poor little innocent crawling ciliate minding its own business, crawling along, and it gets sucked into her tentacle. Now she's not nice, because if she was merciful, she would just kill it, but she doesn't. While it's kicking and screaming and stuck to her tentacle, what she does instead is she shoots a poison into him and paralyzes it. And it paralyzes it so that it can't move. And then she slowly sucks the life out of it. Okay. That's my ex-mother-in-law. And that's me. Mind my own business. She was merciful, she just killed me, but she doesn't. I'm there kicking and screaming. She shoots her poison into me so that I can't move. I had to stand there and listen, and then she just sucks the life out of me. That's my ex-mother-in-law. I always feel better when I teach this class. Okay, this is her in all of her glory. If you look really closely, you can see the actual holes, the suckers in her head. This is my mother-in-law. She got Uncle Joe sucking the life out of him. And she just keep on sucking the life out of him and sucking the life out of him until Uncle Joe can't take it anymore and he just explodes. Okay. So it's not much left. Okay. So they're just like bacteria, there are factors that affect protozoa. And so they're microorganisms, they're affected by temperature, they're affected by pH, and they're affected by nutrients. They're affected by how much oxygen that's in the water too. So we'll see how these things all affect the protozoa. When it's cold, they're slower. When it's warmer, they're more. So you'll see more in the summertime than you'll see in the wintertime. pH, they're more sensitive to pH than the flock formers are. So if you have changes in pH, it might knock out your protozoan. Your flock formers might be still okay, but your effluent, that means, won't be as clean because they're not there swimming around getting rid of all of that extra bacteria that's in the fluid. They need oxygen, just like your bacteria need oxygen. If you lack oxygen, it's going to limit the kind that's there. So the ones that move faster and are more active will start dying off because they need more oxygen, and you'll be left with like amoebas or something like that. Okay, nutrients. Most municipal waters contain the nutrients that they need. It's those industrial systems that are likely to be deficient. Okay, so that's protozoa and metazoa. That's protozoa and bacteria. Both bacteria and protozoa are single cell, and they split into two identical cells. Metazoans are multicellular. They're like us. They're male and female, and the male and females get together and they have eggs. We're metazoans, but we're macroscopic. These are microscopic metazoans. And so they don't do very much at all to clean the wastewater. But what they do is they give us an idea of how old our sludge is. Because some of these metazoans take where your protozoa might take 5 hours or 6 hours to multiply. Some of these guys can take 12, 21 hours to multiply. So if we start seeing these guys dominating in the system, that means your sludge age or your MCRT, your mean cell residence time, is pretty long because you're giving them time to grow and multiply in the system. So the role of metazoans is just to give us an idea of how old or how young our sludge is. They're multicellular. They feed on bacteria, protozoan, algae. They'll even eat any other small metazoan if they can. These are like the meat eaters. These are your rotifers, your nematodes, your gastrotrites, your bristle worms, your water bears, and your ostracods. So the first metazoan to show up on the scene are rotifers. So here's a tidbit. The majority of the rotifers in the treatment system are female. They're both female, but they're both male and female in there, but the females dominate. I wonder do you know why the females dominate? Well, I'm going to tell you why the females dominate. And it's not because they eat the males. The reason why they dominate is because they know how to take care of business, okay, in the treatment plant. So when a female has eggs, the female egg will hatch and she'll go about the wastewater environment and take care of business. But the male egg is called the degenerate. He's literally called the degenerate. So you have to talk to God about that. But he's called a degenerate. And the reason why he's a degenerate is because he doesn't know what to do. All he does is he sits in one spot and he lets out poop pellets. So he just sits there and he craps. And then he waits for a female to come so he can fertilize her, and then he dies. So the whole purpose of the male rotifer is to sit and crap and have sex and die, okay? That's all he wants to do. So that's my ex-husband. And so here is, so their principal contribution is to clean up whatever's left over. Okay, there's 4 different types can be present in the system, and they should never dominate in the system. So this is a female. This is a female. You can tell because somebody said because her mouth is going. This is a jaw. Rotifers have like a mastec, so they can chew. So I was told this must be the female because her mouth is always going. This is the male. He's just sitting there crapping, waiting for his wife to come so he can have sex and die. He's like, when is my wife coming so that I can just have sex and die? So that's a male one. So in the same environment as rotifers are your nematodes. Like these are microscopic worms. These are worms that you cannot see with your naked eye. If you see worms with your naked eye, there's some red worms or brown worms. Those are either midge-fly larvae or sludge worms or tubifex worms. But nematodes are microscopic and you can't see them. And so they come in a variety of sizes and shapes. They move through the fluid. They wiggle back and forth. They'll feed on bacteria, fungi, protozoa. Sometimes they'll eat smaller nematodes. Some of them even have teeth and a spear where they bite and then they shoot the spear in and suck your juices out. This is a normal nematode. This is a normal nematode, just kind of stuck in the sludge. And it wasn't until I moved to Chicago that I realized that this nematode was normal. This is the normal nematode. And then this is a Chicago nematode on speed. I have no idea. This is real time. I don't know what this nematode got a hold of, but this is a Chicago nematode. Okay. And then you have like our water bears. Water bears are commonly found in the same environment as rotifers. They have 8 legs and claws. What the bear will do is take the bottom 4 legs and it will hold on to its prey, and it will take the top 4 and it will just stick the claws in and rip open a hole, and then it will suck all the juices out. And then once it got the juices out and it's got the skin left, then it sucks up the skin. So you see this one, he didn't get all the juice out, so he's still trying to suck up the skin. And so the nematodes water bears are male and female. And they are notorious and able to survive extreme conditions. So what the male will do is the male will take and he will bite the female, and then he will shoot his sperm inside the female. And then he will, once her eggs become fertilized, what she'll do is that she will molt out of her skin, and she will leave her skin, and it will be in the shape of a perfectly shaped water bear, even with the claws, with all of her eggs inside. And then she can have anywhere between 8 to 12 eggs. And those eggs will all be inside the mom's skin. And then each one of those eggs will hatch, and then they'll turn into little bears that go out into the world and kill things. It's hard to tell whether they're male and female without looking at the eggs, because the female leaves it. But if you watch this bear, you'll know this is a male bear because he acts just like a male bear, acts just like a man, and you're going to agree with me that this is a male bear. See, he said, his wife said, honey, I think we lost. And he said, no, we're not. And she said, yes, we are. I don't think we're going anywhere. He said, be quiet. I know what I'm doing. I know where I'm going. That's why I know this is a male bear. This one, though, is a female bear. I don't know. Can you hear this sound? I don't know if you can hear it or not, but she is dancing she is actually dancing to the Nutcracker. Okay, so I'm gonna stop her. But this is the female one with the eggs in it inside. Ooh. So you can see the eggs that's inside. And then this is the eggs that are hatched. And also then there are bristle worms. Bristle worms, they don't do very much to clean the wastewater, but they're just indicators to us whether or not the system is old or not. And then they'll settle out with the sludge, but these are all indicators. You see a bunch of rotifers and nematodes and water bears and all those things, you know that the sludge is pretty old. And this is another bristle worm. And another example. Okay. Okay, and then as you go, as you get longer age systems, maybe like lagoon systems, you'll start seeing water fleas and water mites or something like that. Have you ever seen red fleas on the top of the on your top of your clarifier, like red water fleas skimming on top of your clarifier? Those were probably Daphnia, and the reason why they were red is because they were producing hemoglobin because they were trying to get oxygen. And the reason why they're there is because there's algae there. Algae is their chocolate. Okay. So just real quickly, some things, what we can do. I think we got about a half an hour. What can I do with all of this information I just had? I can look for just little simple things. One thing that I realized, I had the luxury of working at two of the largest plants in the world. 1.8 billion is the Detroit's, 1.2 billion in Chicago. And I had the luxury of being able to just do nothing but microbiology. When I moved to Wisconsin, I worked for the Department of Natural Resources there for 15 years. I realized that the operator, not only did he have to do the operations, he had to do the lab, he had to do the grass, he had to do the snow, and he was the mayor of the city. So he didn't have the luxury to spend time doing just microbiology. So just some quick things that you can do using a microscope. You can look at just something as simple as dispersed growth or the role of bacteria. Just look at how the bacteria are in your system. What you want your bacteria to do is to remove BOD, produce more bacteria, and to form flock and settle. When we see dispersed growth, what that is, that's just a bunch of bacteria hanging out in the fluid, and you shouldn't see that. If your system is operating properly, you shouldn't see a bunch of bacteria in the fluid. So on the right, you should see your flock, and the fluid should be relatively clear. You should never see a bunch of bacteria hanging out in the fluid, because that means something happened. That means something happened. Why is that bacteria dispersed? It means that something interfered with flock formers, or it can also mean like a slug load came in and now the bacteria are all multiplying all over again. Or it could also mean that maybe something came in and knocked out your ciliates or your rotifers, and so they're no longer available to clean that bacteria around. But that's a quick thing. So that means that something interfered with the flock formation. Could be because it's young, that means it's still a lot of food. Toxicity, it affects the enzymes and keeps them from flocking together. A slug discharge, like I said, something knocked out your ciliates, or a bunch of surfactants. Surfactants make water slippery, and so it's hard for things to stick together. So you should never see this. You should never see that in your fluid. You should always look like this. So I want to try to get to the nutrient deficiency before we go. Usually, if you have extremely low food, or if you're deficient in nutrients, you can have some issues with your treatment system. So if I were to take a cross section of a bacteria cell like this, normally your flock formers, they have a fat layer, a lipopolysaccharide layer. They also have a phospholipid layer, a phosphofat layer. And they also have a fat protein layer. Okay, so they have these three layers of fat They need that top layer of fat so they can clump together to form flock. But you need phosphorus in order for them to develop this phosphorus layer properly. They need nitrogen to develop this lipoprotein layer properly. If they're lacking phosphorus, you're just going to get this double layer of fat. If they're lacking nitrogen, you're going to get triple lipo. So you got triple fat. And so you go from having a single layer of fat to having a triple layer of fat. And so where your bacteria would normally clump together to form flock to look like this, then they end up having something like this. And this is an extracellular polymer. So it's viscous, it resists water, it makes it difficult for the sludge to settle, and it can even cause foaming. So you'll see this nice, creamy, milky white substance that's trapped. So all this bacteria trapped in this slime. That's nutrient deficiency. So guess what happens to Daniel when you got bugs that are nutrient deficient? When you got a bunch of fat bugs, he gets a fat head because he has to eat those bugs. So this is what happens to his head. These guys, they ate so much. This is a live shot and they can't even move. It's like we shouldn't have ate the whole thing. You know? And so what happens is you get these nutrient deficient flock particles and you lose settleability and you get foaming. And so instead of your solid settling like this, it settles like this. So this is the same amount of bugs. It won't go down anywhere because you got all this extra polymer in between the particles. And so the solution is to add the deficient nutrient. Ammonia for nitrogen, phosphoric acid for phosphorus, or some kind of fertilizer. If you got some ammonia nitrate or you got orthophosphorus left over after you leave, after it's coming out of the aeration basin, then you're not deficient. But there's one other thing that will make the bugs do this. Besides nutrient deficiency, one other thing will make them do this, and that's the presence of organic acids. Organic acids is a product of anaerobic recycle stream. So if you have an anaerobic digester, or you got you're doing biophosphorus removal, and you got anoxic zones, anaerobic zones, and you got this excess organic acids because you got anaerobic conditions created. That's where organic acids come from. Then this will cause the bacteria to also have that extra slime polymer. So it can be wasted out, but the thing is you need to stop the bacteria from making it, okay? And so I remember one case that I was working on when they had all of that excess slime, excess polymer. What they did is they super wasted. But what they did is they super wasted. The good bugs is the ones that settled at the bottom, and all of the ones that got all the slime was more at the top. So they super wasted, got rid of the good ones, and so you ended up having a whole bunch of slimy bacteria on the top. And so the important thing to do is to slowly waste and replace. And what I mean by replace, now I don't mean necessarily seed. What I mean is make sure that the new bugs that's coming in have sufficient nutrients so that they can start growing and out competing. So you slowly waste, replace, and this will take several sludge ages. And as operators, we are impatient and we want it to happen right now, but it's going to take several sludge ages before it changes. There's also, we can look for evidences of toxicity. Some of these microorganisms have the ability to protect themselves under toxic conditions. And so if half of you guys in the room, if we all went to the Antarctica and half of you guys in the room had on coats and the other half didn't, the ones that have on coats would keep living, but the ones who didn't would die. So that's what happens. Some of these microorganisms have the ability to put on coats to protect themselves under toxic conditions. The ones that can't do that die off, and you end up having a whole bunch of organisms that's got protections, and we call these shells. We call them shells most of the time. So what does bacteria do to protect themselves? Well, there's two types of bacteria that comes in the treatment system: the ones that form flock and the ones that do not form flock. So the ones that form flock, they're within the flock, and so they can kind of protect themselves within the flock. You'll see them secrete some extra polymer to kind of protect themselves in the flock. But what about the ones that's out in the fluid? The ones that's out in the fluid, what they do is they form what's called these zooglio communities. And so they embed themselves in this polymer-like matrix. So and they don't just do it any kind of way. They actually arrange themselves in such a way to benefit the whole. And so they get into these communities, they get rid of their tails, and they say, okay, look, we're going to all join in this community. And they will send out chemical signals, and it's called quorum sensing. They will send out chemical signals and invite only the microbes into that community who benefit the community. In other words, they're not selfish. And so what they'll do is they'll say, okay, you guys on the outer edge, you guys go to the outer edge because you need more oxygen than we do. And we'll just kind of hang out in the center. And then they said, but since you're exposed to the outside, you're exposed to the food and we don't have any. So what you do is you eat food and then you crap and we'll eat your crap. And so the only ones that they want in their community is the ones whose crap they can take. So think about us. We have people in our community who can give us crap because we know them. But some stranger, they couldn't say the same thing to us that our friends could say to us. And so they literally arrange themselves in these communities in a way to benefit the whole. And it protects them against toxic shock. It protects them against changes in pH, drying out organic acids, just being in that community. So you can literally look and see these masses in your sample, in your treatment system as an indicator of stress, because this is a stress response. And so here's an example. Oh, let me go back. So here's an example of just a bunch of bacteria starting to form, mass themselves into this zooglio community. So you got all the bacteria all on the outer edge, let us in, please, let us in. And it's like, no, we don't want to eat your crap. So we don't want to let you in. So they all just kind of embed themselves in that community. And they will embed themselves so tight that nothing can get in. And then once the environment clears up, what happens is the ones on the outer edge will start creating an enzyme that start breaking it apart. And then it'll start getting thinner, and then it'll get thinner. And then eventually little pieces will start breaking apart. And so now all of a sudden your turbidity goes up and you goes, oh my God, it's getting worse. But no, it's not getting worse. That's just the storm before the calm. It just means that it's starting to break up because the environment has cleared. Some form what's called fingered communities. These don't want to eat anybody's crap. So they form in these communities so that the fluid can flow freely between the little channels so everybody can be exposed to some food. Okay. And conditions that affect bacteria also affect Daniel, okay? So if they're embedded in this slimy matrix, your protozoan can't eat them. It's just kind of sitting here on the side of his face teasing him, so he can't eat it. And so you'll also find that your protozoans will start dying off too. This guy right here, he ate so many fat bugs, so he's about to pop. Protozoa also can protect themselves. They can form shells and they can form tubes. And so your amoeba will form shells. And so you'll see something look like this and you'll think, oh, I didn't even know that was an amoeba, but that's an amoeba in a shell. And if you look on the right, you can see the little bit of the amoeba coming out from underneath the shell. Here's another example of a shelled amoeba. Some of the amoeba can take and use your sludge to make a shell, and some of them will secrete and make their own shell. But when you start seeing a bunch of shelled species, that means that environment have somehow become harsh. Some low levels of toxicity or something, not enough to kill them, but enough to make them put on some protections. Here's not a Krispy Kreme donut, but this is a shelled amoeba. And you see them, it's a bunch of them. And so you can see the amoeba kind of oozing out of the shell. Another example. And that's another example of a shelled amoeba. Some of them look like pineapples. Flagellates also can protect themselves. What they do is they do just like the bacteria do. They embed themselves in a gelatinous matrix. And so they kind of look like this. You'll see them embedding themselves. The big flagellates, they just die. The little ones can embed themselves and protect themselves against harsh environments. All of these are just indicators that something harsh has hit your plant. See, he's going to be brave and think he's going to go out and test them. His wife said, wait, babe, I'm going with you. And they both die together. Okay. So stalk ciliates, what does stalk ciliates do when under unfavorable conditions? Well, they dislodge themselves from their stalks. So if you were gentle when you made your slide and you didn't knock the heads off yourself, then, and you look at a sample and you see a bunch of headless stalks, that's a sign. Because what stalk ciliates will do is the heads will dislodge and they will swim away and try to find a more favorable area. If it finds a favorable area, it will grow a new stalk. And if you can tell this one, because if you look and be kind of on the, over here, you can see sort of the milky white area where the bacteria is protecting itself too. Some form tubes. So some of them will, some of the attached ciliates will form a tube. So it will come out of its tube, fill up its body, fill it all the way up, and then it will contract itself back inside the tube. So if you see a bunch of I call them tube dwellers. So if you see a bunch of tube dwellers, you see a bunch of shelled amoeba, you see a bunch of masses of zooglea, you know something toxic hit your plant. Some of them have single tubes, and then some of them share a tube. It's two different species. Rotifers also form shells. This is a shelled rotifer. So if you've ever seen a rotifer that kind of scrunches itself up like that, this is a shelled rotifer. It's like a turtle shell. These are all indicators of toxicity or harsh conditions. Another example of it. Water bears can also protect themselves. Water bears can do what's called cryptobiosis. So they can dry themselves up into a husk or a powder. Some of us who are old, remember back when they had the sea monkeys. The sea monkeys are brine shrimp, and brine shrimp can do the same thing. They can dry themselves up to a powder, and then when you put them back in water, they come back to life. But they have found husks of water bears that's been 30 years old and have been able to rehydrate them, and they come back to life. So there are other indications of toxicity. If you look and you all of a sudden you don't have any higher life forms like any kind of protozoa or metazoa, that's an indicator something harsh is coming in. Poor flock formation, low oxygen use, dead things don't use oxygen. Poor BOD removal, dead things don't remove BOD. A flagellate bloom, because you remember, like I told you, flagellates like to eat the juices of dead things. Or if all of a sudden you come in and you don't have any more filament, you have filaments all of a sudden. Filament is bacteria. A lot of times after a toxic event, filaments have the ability to recover faster than the other microorganisms. The important thing is to keep a process chart of your treatment parameters and measure them routinely and consistently, and do more than just what's required by your permit. I really like this cartoon because it says, he goes to the doctor and he says, doctor, I got a broken arm, and he says, but can't you just fix it in Photoshop, on the computer? And he's saying, yeah, I can fix it in Photoshop, but it's still going to be broken. And so that's what we do. A lot of times at our plant, we'll throw in some chlorine. It's still broken. It might look like it's better. We'll add some polymer to help it to settle, but it's still broken. And so what we want to do is we want to get in the habit of finding out what it is that happened to our plant that caused it to break. And we fix that so that we don't have to fix it into Photoshop. And so with that, I am done. And if you guys have any questions, I'll be glad to answer.
Any questions? Put your hands up. We'll bring you the mic. Any online? Okay.
I know that was a lot in that short time. Sorry.
Here comes one.
Thank you, guys.
Hi, nice presentation.
Thank you.
I was going, by the middle of the presentation, I was thinking, I hope it covers the food nutrient nitrogen phosphorus ratio for monitoring a lagoon system. In our city, we have lagoon systems. And then you start talking about what potential rotifers and microorganisms in lagoon systems could happen. So that question is covered. But yeah, I still have the question about what the food nitrogen phosphorus ratio for lagoon systems would be.
It's the same because that's just a natural thing for microorganisms, for bacteria they need. But for lagoon systems, that's like a whole different class because that's a whole different set of microbes. You've got the different zones. You've got your anaerobic zones. You got your facultative zones, and then you got your aerobic zones. Then you got your algae, and then you got a succession of different types of algae. So it's a lot more complex systems than activated sludge, but the nutrient ratio is still the same.
Still the same. Great. And very specific question, if we would like to start looking at the microbiological population in the lagoon system, what type of equipment do we need, like microscope, what size and so on?
Well, you want to make sure you have a phase contrast microscope because because water is clear, microorganisms clear. Regular bright field microscopes don't show really good structure. Phase contrast, what it does is it makes the difference between the liquid and the microorganism more distinct. You can get a good phase contrast microscope for under $3,000. And you get even some cheaper, but you want to make sure it has what's called phase contrast, and you want to make sure you have 100x objective, which is called an oil immersion lens, because you want to be able to multiply. Because you're going to be looking at bacteria or algae or protozoas. You want to be able to magnify it 1,000 times to see some of the bacteria and stuff.
Excellent. Thank you.
You're welcome.
One in the back over here.
Great. Now I don't want to drink my glass of water.
Just going back to the microscope, I'm wondering if you have any training courses that you'd recommend for learning how to use the contrast and just all the different features.
My course, I have online courses, but I also have do a in-person on-site where everybody bring their microscope and bring their samples, and we look at the samples and we actually do some counts and some quantifications and all those things. And so if you guys have my information, you can look at my website or shoot me an email, and I can give you further information.
Awesome, thank you.
Or I can give it to EOs. You give it to them, and it'll be available to you through them.
That sounds good, yeah. Any online? Nobody? Yeah, I think everyone had a real good time with your presentation.
Well, and not everybody are microbiologists, so I think you remember my family better than you would if I called it Cartesium or Percolarium or something like that.
So if we got nothing else, no other questions, let's just give a big round of applause here for Tony. Thank you so
guys.
much.
Thank you.
And yeah, hope to have you again next year.
Okay.
Everyone really enjoys it, so thank you so much.
Okay. Goodbye, everybody.
Bye-bye.